We ended the last lecture by asking what would happen if a nerve cell's membrane suddenly became much more permeable to sodium ions. If sodium could flow into the cell more freely, based on what we know, we'd predict that the voltage across the membrane would be driven to a much more positive value as those positively charged sodium ions rushed into the cell. This is exactly what happens when a neuron becomes active, that is, when it fires what we call an action potential. Let's explore how this works a little bit by looking at what would happen if we experimentally perturbed the membrane potential of a neuron. Now, we could do this, for example, by actually putting an electrode into the neuron. Now, last time we put an electrode into a neuron, we used it to measure the voltage across the membrane. But this time, we're actually going to use this electrode to add positive charges. In other words, we could run what we call current in or out of the cell through that electrode. Current simply means the flow of positive charges. And as this current, we would use positively charged ions, like sodium ions. Now, because the membrane potential of a neuron at rest is negative inside with respect to outside, we say that that membrane is electrically polarized, or a polarized membrane. If we, with our electrode, increase the flow of positive charges into the cell, we'd be adding positive charges into the cell, positive ions, and we would cause the inside of the cell to become a little bit less negative. We would say that the cell membrane has become depolarized. Now, if we were looking at this on our voltmeter, what we would see is that as we added positive charge to the cell, the voltage would go closer to zero. Say it would rise from minus 65 millivolts to maybe minus 60 millivolts. Now, the interesting thing is the more current we add, the more positive charge we put into the cell, the more we can make the voltage become more positive, closer to zero, the more we can depolarize the cell. So, for example, we could add enough current to make that cell membrane go from minus 65 millivolts up to minus 55 millivolts, or even farther. But what is really interesting is that now that we've got our ability to put current into the cell, if we add enough current to drive the cell membrane to a sufficiently high level, say maybe about minus 50 or minus 45 millivolts, all of a sudden something else happens. Without us doing anything, the cell membrane will all of a sudden radically change its voltage. The cell membrane actually will shoot way up to have a very positive value, say as positive as about minus 50, or positive 55 or even positive 60 millivolts inside relative to outside. What's happened is that the initial current that we've added to the cell somehow triggered a series of events that changed the whole relationship of the nature of the voltage inside versus outside the cell. And what we want to understand is what it was on a molecular level that was responsible for that change. Well, one other thing I want to point out about this event that we've just triggered artificially by adding current ourselves to the cell is that it occurs it occurs in the same way each time. In other words, if we put enough current into that cell to drive it to a high enough positive value, what we would call the threshold value, then when that cell goes very positive, it will always go up to the same value. We refer to this as an all or none event. In other words, it happens the same way each time. And there's one more thing I want to describe about this action potential, and that is that after it rises to a very positive value, it will almost immediately, within a millisecond or two, drop back down. In fact, it'll drop back down to a negative value, a value that's even more negative than the resting membrane potential normally is. And then after a few more milliseconds, it will come back and it'll be at its resting value. So let's review what we've just seen. We've added some current to the cell. We've caused the cell to become more positive on the inside, just a little bit. If we add enough to cause the cell to pass a threshold, then all of a sudden an action potential occurs where the cell membrane potential rises to a very positive value and then rapidly drops back down to a very negative value and eventually comes back to its resting value. What's responsible for this? Well, the action potential results from the rapid and reversible change from a series of, I should say, rapid and reversible changes in ionic permeabilities, specifically permeabilities of sodium and potassium across that cell membrane. These changes in permeabilities reflect the activity of gated ion channels, and specifically ion channels that are gated by a change in voltage. These gated ion channels are normally closed when the cell membrane is at rest, but they open up when the cell membrane changes its polarization, becoming sufficiently depolarized that some threshold is passed. Now let me tell you a little bit more about what we would call voltage-gated ion channels in a neuron cell membrane. There are really two types of voltage-gated channels we need to think about here. One is a selective channel for sodium, and the other is a selective channel for potassium. So we have a selective voltage-gated sodium channel and a selective voltage-gated potassium channel. Now these channels share in common the fact that they will open up when the membrane in which they find themselves becomes sufficiently depolarized. But they differ in two very important ways. First, the sodium voltage-gated channel responds very quickly to a change in the membrane voltage, in other words, very quickly to depolarization. By contrast, the potassium voltage-gated channel only responds more slowly. What I mean by that is that if you depolarize the membrane past a threshold, the sodium-gated channel will open up right away. The potassium channel takes a little more time. Now, a second important difference between these two kinds of channels is that the sodium voltage-gated channel only stays open for a short period of time, regardless of what the membrane potential is. In other words, it opens up in response to a depolarization, but once it's open, it only will stay open for a millisecond or two, and then it just snaps shut, regardless. By contrast, the potassium voltage-gated channel will be open when the membrane is sufficiently depolarized, and it won't close until the membrane goes back to some more negative value, closer to the resting membrane potential. So we've got these two very specialized gated ion channels that are gated by changes of voltage. Now, armed with this background, let's take a look at how these two voltage-gated channels collaborate to produce an action potential. Now remember, an action potential, if we just were looking at an action potential on our voltmeter, or actually if we looked at that action potential on an oscilloscope where we would see a change of voltage over time, we would see at the beginning of the action potential the voltage would rise to a very positive value, then it would very rapidly fall down to a negative value, even a little more negative than rest, and then it would slowly, in a couple of more milliseconds, come back up to its resting membrane potential. At the resting membrane potential, both the sodium and potassium voltage-gated channels are closed and won't permit ions to cross them. Now, when the membrane begins to depolarize, for whatever reason, and remember, in our experiment, we depolarized it ourselves by adding some current through our electrode. When the membrane begins to depolarize, this causes the voltage-gated channels to begin to open once the membrane is depolarized sufficiently, reached its threshold. Now, the interesting thing here is, remember, the sodium voltage-gated channel opens very quickly. So when the membrane depolarizes enough, all of these sodium voltage-gated channels snap open, and all of a sudden the floodgate is open to sodium. And remember, sodium wants to rush in the cell. It's being pushed up down its concentration gradient into the cell, and also down an electrical gradient into the cell, or it's being pushed from positive to negative into the cell. So these forces both are pushing sodium to the cell. Sodium, when all of these voltage-gated channels snap open, is now 20 times more permeable than potassium was. Remember, at rest, potassium is about 20 times more permeable than sodium. What this means is that sodium dominates what the membrane potential will be. And because sodium is a positive ion and is rushing into the cell, the membrane potential quickly rises to a more positive value, shooting way up to about plus 55 or plus 60 millivolts. Now, remember also that these sodium voltage-gated channels don't stay open very long. They only stay open a millisecond or two. So what that means is that very shortly after all these sodium voltage-gated channels have been opened, they snap shut. All of a sudden, the permeability of sodium comes crashing down. And at about the same time, not exactly, but also what's happening is that those voltage-gated potassium channels, which are slower in responding to the depolarization, now begin to open. So what's happened is that we've reduced the permeability to sodium because those voltage-gated channels are snapping shut, and we've even increased the permeability to potassium even more because now all of these new potassium channels, the voltage-gated ones, open up. Well, what happens then to the movement of these ions? Well, now potassium is far more permeable across the cell membrane, and sodium is not so permeable. And that means that positive charge will now be pushed outside the cell. Potassium will be pushed outside of the cell very rapidly. And the movement of positive charge from the inside to the outside causes the membrane potential difference to now crash down to a very negative value again. In fact, because there are so many voltage-gated potassium channels open in addition to the normal passive ones that we have at rest, the membrane potential crashes down to an even more negative value than at rest. We call this part of the action potential where it becomes more negative than rest, the undershoot. Well, eventually, because now these potassium channels find themselves, the voltage-gated ones, back in a polarized membrane, which is below the threshold, these voltage-gated potassium channels will eventually close down as well. Again, they close down more slowly, just as they open more slowly, and within a few milliseconds, only the passive channels we talked about in the last lecture remain open, and the cell membrane goes back to rest. There's an interesting point to make about this process by which the action potential is generated in the context of what we referred to a couple of lectures ago as negative versus positive feedback. The action potential is an example of a positive feedback system because the initial depolarization, at least with respect to the activity of the sodium channels, the initial depolarization caused these voltage-gated sodium channels to open up. When those voltage-gated sodium channels open up, what that means is that there's even more positive current rushing into the cell, which causes even more voltage-gated sodium channels opening up, and you have a positive feedback loop, which is why the action potential is an all or none event. Once it crosses the threshold, once the cell membrane's potential crosses the threshold, then the opening of the potassium channels will continue till completion, and you will always go to the same positive value. Well, there's one last important thing to add to our understanding of how action potentials work, which will become important in just a minute, which also is a result of how the sodium voltage-gated channels work in specific. After an action potential occurs in a neuron, there's a refractory period, during which time another action potential can't be triggered. So let's go back to where we started with our own electrode in the cell where we can inject positive charge. And let's say we've injected positive charge, and that's what depolarized the membrane past its threshold and caused an action potential. Well, the action potential causes itself, the membrane potential, to shoot up to its peak, and then the membrane potential comes crashing back down to a negative value. Let's say right there at that point, we added more current. We tried to depolarize the cell past its threshold again. Well, there'll be a certain period of time, a number of milliseconds after the cell has fired its action potential, where we can no longer trigger another action potential. The cell has gone refractory. The reason for this refractory period has to do with the way that the sodium voltage-gated channel is thought to work. Remember, the sodium voltage-gated channel opens up in response to depolarization, but then it just snaps shut again. Well, it turns out that it seems as though there are two different parts of this protein that change shape. There's one part of the protein that changes shape in response to a voltage difference across the membrane. That's the gate, as it were, that opens up in response to depolarization, opening up the sodium channel. But there's another part of the protein that, as a consequence of that first change in shape, will change its shape in such a way as to close the channel. This is what we call the inactivation gate, because it inactivates the sodium channel. Once the inactivation gate has swung shut, as it were, on this protein, it stays shut for a certain period of time regardless, a number of milliseconds before eventually the protein will return to its resting configuration and the inactivation gate opens up. The refractory period of a neuron following an action potential is the result of the fact that all of the sodium voltage-gated channels in that neuron's membrane have their inactivation gate shut, and they'll remain shut for a certain period of time. So there's nothing we can do to trigger another action potential because we can't get those sodium gates to open up again. Well, the action potential reflects a very orchestrated, a highly choreographed interaction between ion channels that creates this very specific and regular change in the membrane potential of a cell. And that change in the membrane potential, the action potential, is really the basic currency of information of neurons. A neuron will fire an action potential or not, and when it does so, it will fire it the same way each time. If we draw an analogy to the way that computers work, it's as though the neuron has either an on state or an off state, a 1 or a 0. An action potential is firing or it's not firing. And this is a very useful way to process information. But there's another part of the system that we haven't actually talked about yet. Because remember, this information is only useful. The action potential really is only useful if it is able to go from one place to another. And up to this point, we've been talking about an action potential just occurring in a single patch or a single, just like in a round neuron-like cell. What does this action potential do for us once it's generated? Well, obviously, what the action potential has to be able to do for us is to propagate from one part of the cell to the other. And that's what the axon is all about. Because what happens is once an action potential is triggered, say in the cell body of a neuron, that action potential will travel into different parts of the neuron and especially travel down the axon. Now, how is it that, and there's one more point I want to make about that, and that is that every time this action potential travels down the axon, it does so again in an all or none fashion, in the same way every time without any degradation. So if we measured an action potential at one end of an axon and then measured the action potential in that neuron at the other end of the axon, they would look the same on our oscilloscope. There's no degradation. And that also is very important for this information to be able to be transmitted to different parts of the body. Now the question is, how does this action potential travel down what's essentially this cellular wire? Well, the answer to this question really is quite simple. Let's consider now an isolated length of an axon. Imagine that we've got an axon of a neuron and we're looking at the two ends of it, like looking at two ends of a cellular tube. And imagine that we somehow trigger an action potential on one end of this length of the axon. What happens to the depolarization that we cause or that is caused when that action potential is triggered? Well, as a consequence of the action potential that's been triggered, let's say we've put an electrode at one end of that axon, we know now that sodium will rush into the cell because the permeability of sodium greatly increases where that depolarization has occurred because the voltage-gated sodium channels have begun to open. Now, this inflow of sodium not only depolarizes the immediate area that not only further depolarizes the immediate area where the original depolarization occurred, that is where the voltage-gated sodium channels are opening, but the sodium will flow in both directions down that cellular wire. In other words, sodium ions will rush into the cell wherever the voltage-gated channels have begun to open, but then flow to adjacent areas. So you can imagine that what happens is that the adjacent area of the initial patch that's depolarized will itself begin to depolarize and essentially become what we call recruited to an action potential. If we imagined this cellular wire as a series of discrete patches, it's as though one patch depolarizes, some of the depolarization leaks to the next patch, which causes that patch to depolarize enough beyond threshold, which then leaks to the next patch and so forth. Now, of course, it's really not a set of discrete patches, but a continuous process. But you get the point. It's as though there's a wave of depolarization that flows down the axon as an action potential is traveling from one end of the neuron to another. What's interesting is that in neurons, action potentials only generally travel one way. In neurons in organisms, action potentials usually originate at or near the cell body, and they travel down the end of the axon to the synaptic terminal, which will be adjacent to the next neuron down the line in the system. So you might ask, why is it that this action potential only goes in one direction? Because the model I just drew for you would suggest that the action potential actually could go in both directions. We have a patch of this piece of the axon. It's depolarized. We know that the positive charge that flows into that patch of the axon will flow both in one direction and in the other direction. Why doesn't that depolarization travel normally in both directions? Well, the answer to that question has to do with the fact that these sodium voltage-gated channels inactivate. Remember, after a patch of a neuron has undergone an action potential, the sodium voltage-gated channels in that area now are going to remain shut because their inactivation gates are closed. So if an action potential starts at one end of the axon and starts moving down the axon, at any one point in that process, sodium that is rushing into the cell will flow both in the direction that the action potential is moving and back to where the action potential came from. But the voltage-gated sodium channels that are in the backwards direction will be inactive. And therefore, that patch of the membrane of the neuron will be refractory. So we have only the progression of the action potential in one direction. And it turns out that the morphological specializations of the neurons are such that the action potentials that are generated in one neuron are generated at or around the cell body. Often they're actually recruited right at the base of the axon, a part of the axon that is sometimes called the axon hillock. And then once an action potential is generated at the base of the axon nearest to the cell body, it will only propagate away from the cell to the next neuron that it's going to contact. Well, what have we accomplished up to this point? We've now considered two of those special properties of nerve cells that I outlined at the beginning of the last lecture. We've seen how an action potential is generated, that is, how we get this highly specific and highly orchestrated change in the cell membrane of a neuron, called the action potential. And we've also seen how this action potential propagates, specifically propagating along these elongated axons. Let's turn briefly to the third special property I listed, which is how the information of one neuron is conveyed to another neuron, or to put it another way, how an action potential in one neuron can potentially trigger an action potential in another cell. Now, as I said, typically the action potential will travel from a cell body down to the end of the axon to its synaptic terminals, which are in close juxtaposition to another neuron. These axon terminals form what we call a synapse on the other neuron. And so what we want to do is to essentially ask, how do synapses work? How does the mechanism that connects one neuron to another work so that some sort of electrical event in the first neuron causes an electrical event in the next neuron? Now, just to keep things straight, I want to give you a couple of terms here. We call the cell that's transmitting the action potential, when we're thinking about a synapse, the presynaptic cell. And we call the cell that's receiving the information, if it does, the postsynaptic cell. So in our model of two neurons, we would imagine that the axon from one neuron goes and makes connections with its synaptic terminals on the cell bodies and dendrites of another neuron. The first neuron we will call the presynaptic neuron, and the second neuron we'll call the postsynaptic neuron. Now, given that the action potential is an electric signal, you might guess that what a synapse is a specialized structure that allows the depolarization that's occurring in one cell to just pass directly into an adjacent cell. And indeed, there are some kind of synapses that work this way. We call these electrical synapses. And in fact, what electrical synapses are very, very large non-selective ion channels that are located on both the presynaptic and postsynaptic sides of a synapse in alignment, so that the two channels line up, forming a continuous channel from one cell to another. And in an electrical synapse, what we can have then is the depolarization just passing from the presynaptic side to the postsynaptic side of the synapse. It turns out, however, that electrical synapses are relatively uncommon. In fact, they're mostly found in invertebrate animals and only very rarely found in vertebrate animals such as ourselves. By far the most common kind of connection between nerve cells isn't an electrical connection directly, but instead a chemical one. In a chemical synapse, there's a very narrow specialized space between the membrane of the presynaptic cell's axon terminal and the membrane of the postsynaptic cell. This is called the synaptic cleft. The depolarization that is occurring on the presynaptic side of these two cells together can't directly cross this space. Why not? Because this space is separated by a small gap in two cell membranes. The depolarization can't just simply cross that. Ions can't cross the cell membrane. Instead, what happens, really through a remarkable sequence of events, is that the electrical signal in one cell, the action potential arriving at the synapse, the synaptic terminal, is converted into a chemical signal. The chemical signal crosses the small space between those cells, the synaptic cleft, and then that chemical signal is converted back into an electrical signal. Let's look very briefly at how this works. It actually involves the same kinds of cell signaling mechanisms that we've now seen in a couple of contexts. If we could look up close at one of these synapses, get a really good electron microscope and work out the anatomy in some detail, you'd see that the presynaptic side and the postsynaptic side of a synapse look quite different. On the presynaptic side, on the cellular level, you would see a large number of vesicles in the cell right near the edge. These vesicles, which are the kinds of vesicles that are produced by the Golgi apparatus, as we discussed earlier, are filled with neurotransmitters. Remember, neurotransmitters are those chemicals specialized for chemical signaling between neurons. There are lots of different kinds of neurotransmitters specialized for different kinds of nerve cells, but by and large, we can all consider them a kind of chemical signal. And they're stored in vesicles on the presynaptic side of a synapse. When depolarization arrives at the presynaptic side of the synapse, that is, when an action potential propagates down to the end of this axon to its synaptic terminals, that depolarization causes those vesicles to move to the cell membrane, fuse with the cell membrane, and to release the neurotransmitter into the synaptic cleft. Now, the way that this works is actually interesting and complicated, and we don't have time to go into it here, but by and large, it involves a second messenger, calcium ions actually, coming into the cell through their own set of voltage-gated channels when the presynaptic side of the cell depolarizes. As calcium rushes into the cell, the calcium as a second messenger triggers a number of molecular events that move the vesicles to the membrane, cause those vesicles to fuse with the membrane, and release their chemical contents into the cleft. In this way, the electrical signal has been transduced to a chemical signal. The action potential has caused the release of neurotransmitter specifically at this specialized connection between two cells. Now, if we looked in detail at the postsynaptic side, that is the membrane of the cell that's receiving the signal, what we would see are receptors that were specific for that neurotransmitter, transmembrane receptors that would actually specifically bind with the neurotransmitter on the extracellular surface, and using the same kinds of cell signaling mechanisms we saw before, cause changes on the inside of that cell, which would, through second messenger systems or a variety of other mechanisms, open up gated ion channels. Now, these gated ion channels in this case are chemically gated, or we might say ligand-gated channels. These are channels that are opening up because of the binding of the neurotransmitter that was released on the presynaptic side of the cleft that has moved to the postsynaptic side of the cleft. When the neurotransmitter binds on the postsynaptic side, this triggers, through a variety of mechanisms, ligand-gated channels to open, and those ligand-gated channels then allow sodium or other positive ions to rush into the postsynaptic cell, depolarizing that cell. If a sufficient amount of depolarization rushes into the postsynaptic cell, it itself will fire an action potential. In general, a single chemical synapse isn't sufficient to depolarize the cell enough so that it will actually fire an action potential. And in general, a postsynaptic neuron will have many, many different synapses made on it by one or many, in fact, in some cases, thousands of cells. And it's the collaboration of the depolarization of all of these synapses that will make that postsynaptic cell either fire or not fire an action potential of its own. Well, we've only begun to really look at how the nervous system carries physiological information. We've really only begun to see how an electrical signal can be generated and propagate and induce a chemical signal to be released that will essentially trigger or not trigger an action potential in another cell. And this is just scratching the surface of asking the question, what is physiological information? But at this point, I think it's worth pondering for a moment on the implications of what we've learned so far. The remarkable ability of neurons to process and to transmit information essentially all boils down to changes in the permeability of a few ion species, notably sodium and potassium. Now, if you think about that, it means that things like the trick of juggling, or maybe more complex things like the emotion you have when you see your wife or your mother, or fear, or joy. All of this is something that's happening in our nervous system. And all of this is due to the activity of ion channels opening and closing. I actually think that's a profound realization, one that's both exciting and challenging, and I think one that provides an interesting and tangible interface between the realms of biology and psychology and even philosophy.